Good afternoon. The human brain is a powerful thing. And as biologists, you sit here and you think, how cliche is it to say that? We know the brain's powerful. We know what it's capable of. But did you know that every single day, your brain is making connections or breaking connections based on the things that you do? Things like how much sleep you get, how stressed you are, the amount of exercise you get, how much you study, your diet. All of these things can affect the making or breaking of connections in your brain. Making connections is a concept known as neurogenesis. Neurogenesis is the formation of neurons or branching of neurons. Neurogenesis is important to scientists and they study the proteins involved. So why is it so important? Why is what's normal what we study? Well, we study the normal function so that when things aren't normal, we can know what went wrong. When things aren't normal, oftentimes it's the result of a neurodegenerative disease, which I'm going to refer to as NDD. And NDD is when you lose your neural functioning, when the branches stop growing or when they even break down. Neurodegenerative diseases affect so many people. And you know someone, I'm willing to bet, or if you don't know someone, you know someone who does know someone personally who has an NDD. And if you're one of those people who knows someone with an NDD, you also know that treatment is next to impossible. And there's no cures. And that diagnosis is ineffective. So if you had someone in your family with Alzheimer's disease, if you wanted to get tested to see if you could develop Alzheimer's disease, it's not going to be the most accurate test. So studying these protein pathways is a way to develop a new test or a new treatment to help cure these diseases or to help you discover early if you could develop them. So we study these proteins, and Pichette and Piccoli actually came up with a new pathway. So where did they start? How did they find these proteins? Well, they looked back on different research, on old research, and they found a supergroup of proteins called immunoglobulins. Immunoglobulins are a superfamily of proteins. You can tell just by looking at the name and the prefix immuno that they are antibodies. They're found all over the body, and they're involved in cell adhesion and recognition. So now that they have this huge supergroup of proteins, they need to narrow it down some. So again, they looked to research, and they found that IgLON proteins are a subgroup, and that they've been shown to influence neurogenesis. And because IgLON proteins show neurogenesis, it's a perfect proteins for them to use. So once again, they narrowed it down some to a protein in the IgLON family. NEGR1 is a protein that they studied. And because we know it's an IgLON, and because we know it's in the immunoglobulin superfamily, we know it's anchored to the lipid membrane, we know its influence in neurogenesis, and we also know that it's involved in cell adhesion and recognition. So Pichette and Piccoli found a potential mechanism by which neurogenesis is happening based on the NEGR1 protein. How did they do it? Well, they had several questions. And so what they did was they took primary neural cultures and they treated them all similarly, but then they gave them different treatments based on what they were looking for. So first they targeted FGFR, which is a growth factor receptor, and then they targeted NEGR1, and then they cloned them into a lentivirus, GFP expressing PLVTH. Once they did that, they transduced the neural cultures from mouse embryos with the lentivirus. After they transduced these cultures, they then gave them a treatment based on what they were questioning. Once that was done, they looked at the positive neurons with the GFP, and then they used a Bradford assay or a Western blot to purify and detect the proteins there. Then they measured the proteins and traced them based on their length and the number of branches on the neurons. So what were their questions? Their first question was pretty straightforward. Does NEGR1 in fact influence branching of neurons? How they did this was they took primary neurons and infected them with high and low concentrations of siRNA that was targeted to NEGR1, or they treated them with a control, which is referred to as either siNEGR1 or si control. siRNA is RNA that is specifically set up to target something and break it down. So it's an inhibitor. So in this case, siNEGR1 silenced NEGR1. So what they found was that neurons silenced had a lower count of branching and a shorter length. And you can tell this by their pictures of their tracings, that in high and low treated SENEGR1 proteins, when they were silenced, they had less branching, just by seeing it here. But then they had a graph, and it measured the amount of neuron branches and the length. And you can tell with high and low concentrations that it seems to be dosage dependent. And their little figures up here represent significance. And if it's an asterisk, it's a p-value of less than 0.01. If it is a pound sign, the p-value is less than 0.05. So obviously that there is a significant difference between high and low concentrations of silenced NEGR1. So it seems like it's dosage dependent. So then they led to further questions. And they found that there was NEGR1 in the media with another protein called NCAMP. And NEGR1 shouldn't be in the media because it's bound to the membrane. It's a membrane-bound protein. But NCAMP is shed by atom 10, which is a sheddase. And NCAMP is an immunoglobulin. So they thought, could atom 10 also shed NEGR1? So their second question was, is NEGR1 able to work as a soluble protein after being shed by atom 10? So what they did was they treated primary neurons with either a control of dimethyl sulfoxide or an atom 10 inhibitor. The atom 10 inhibitor is named GI-254-023X, and it's abbreviated in their graphs as GI. So what they found was that neural growth is reduced when atom 10 is inhibited. And this led to another question: what is the contribution of soluble NEGR1 to the branching of neurons? They know that atom 10 cleaves NEGR1 based on what they just found out. And so now that it works as a soluble factor because it's released from the membrane, how does it work as a soluble factor? So DMSO and atom 10 inhibited neurons were both treated with purified NEGR1. Purified NEGR1 is abbreviated as rNEGR1, and it can act as a soluble factor. So once they treated them with the purified NEGR1, they found that neural growth was rescued in the things that had been atom 10 inhibited. And you can tell by the pictures here, this is the one that has been treated with the inhibitor and the purified NEGR1, and this is just the inhibitor. There's obviously more branching. So this led to more questions. What proteins does soluble NEGR1 bind to encourage neural growth? Well, they looked back at research, they looked back at old papers, and they found that there's a lot of proteins that influence neurogenesis. So they looked at 3. The proteins they studied were membrane-bound NEGR1, because they know that NEGR1 is cell adhesion, so it can bind to itself. So they wanted to know if making a homodimer was needed to influence branching. Then they looked at the ERK1/2 kinase, and it is a pathway that triggers a cascade of events. Then they looked at the growth factor, FGFR2, which is involved in cell division and cell growth. The first question was, does soluble NEGR1 bind to membrane-bound NEGR1? So what they did was they treated primary cultures with siNEGR1 and the control, and then went back and gave them purified NEGR1. What they found was that cultures treated with the silenced NEGR1 had impaired growth, like they expected. But then adding purified NEGR1 was able to rescue the branching. So with this information, they could tell that it didn't need to make a homodimer. So, because siNEGR1 silenced NEGR1 at the membrane, and purified NEGR1 came in as a soluble factor and was able to rescue the growth, as you can see here, as compared to here, it doesn't need to bind to itself, and that's not necessary for neurogenesis. So then they said, can NEGR1 influence the morphology of neurons through the ERK1/2 pathway? ERK1/2 is a kinase that's responsible for signal transduction. So if you look at the pathway, a growth factor binds to a receptor tyrosine kinase, and then it goes through RAS and RAF and MEK1/2, and that phosphorylates ERK1/2, which then activates Krebs. So what they did was they inhibited the MEK1/2 right here. So the control and our NEGR1 treated cultures were then treated with a MEK inhibitor. And what they found was that purified NEGR1 was not able to rescue the branching of neurons treated with MEK inhibitor. As you can see here, this one was inhibited and this one was inhibited and given purified NEGR1. So it wasn't able to rescue it. So this suggests that NEGR1 activates a pathway that allows ERK1/2 to be phosphorylated. Then, is there a correlation between NEGR1 and FGFR2? FGFR2 is a receptor that they think could be the receptor that activates the ERK1/2 pathway. How they did this was primary neurons were infected with SIFGFR2, or a control, and then they were given purified NEGR1. SIFGFR2 works the same way siNEGR1 does. It's RNA targeted to inhibit FGFR2. So, when they treated it with the purified NEGR1, they found that NEGR1 was not able to rescue the branching. So with this said, even though they gave it purified NEGR1, NEGR1 must be dependent on FGFR2 to work. So based on what they found, NEGR1 probably binds to this receptor, and that receptor is probably responsible for activating other pathways. Based on the other things they found, they came up with a conclusion. The conclusion was they found a new pathway. So what happens is atom 10 at the membrane sheds NEGR1. NEGR1 then goes and binds with FGFR2, which is a receptor. That goes through this kinase and phosphorylates MEK, which phosphorylates ERK1/2, which then goes and activates Krebs, which stimulates neurogenesis. So now that they found this new pathway, they found a new target protein. They know what this protein does, so they can have a new diagnosis. They can check to see your levels of NEGR1. They also know that this protein influences neurogenesis, so that could lead to possible treatments. And even though this is a fairly new study, it still provides valuable information to the field of neurogenesis and the field of medical research. Thank you.